After studying about transcription and before starting translation, it is necessary to understand few terms like genetic code. Genetic code can be defined as the sequence of nitrogenous bases that can be called even as nucleotides in mRNA, which contains the information for the synthesis of new protein molecules. Therefore, genetic code designs the information that is present in DNA that has to be transcribed and carried by mRNA to the cytoplasm for the protein synthesis. Thus, genetic codes designs or creates the information in DNA that is present in DNA, which is transcribed and is carried by the mRNA that is messenger RNA to the cytoplasm for protein synthesis. Thus, after the formation of mRNA and before getting converted to proteins, that is before the synthesis of proteins, the knowledge of genetic code is very essential because it is this genetic code which designs the information for protein synthesis. Now, as we all know that proteins are synthesized by DNA, therefore, a DNA strand in a chromosome represents many genes. Each gene on this basis can be represented as a part of DNA molecule and therefore have its unique sequence of nucleotides. Thus, a gene that is a part of DNA have its unique sequence of nucleotides in that particular length of DNA. And therefore, it is this information that is present in this sequence of nucleotides occurs in the form of genetic code, and so here genetic code plays an important role for designing the information. As a result of this, each gene may contain thousands of based unique sequences. Now, how is this genetic code within the DNA described or occurs? In order to understand its structure, that is genetic code arrangement, it is necessary to understand the triple code. Now, triple code means the arrangement of nitrogen bases within the genetic code that constitutes and provides the information for protein synthesis. Till now, the main problem of genetic code was to determine the exact number of nucleotides that are present within a gene. And helps in protein synthesis, and thereby that codes for one amino acid. As studied earlier, in nature there are about naturally occurring 20 amino acids. So, in order to study the genetic composition of such amino acids, it is necessary to understand the genetic code structure within them. As said earlier, there are mainly 4 nitrogenous bases, that is, mainly 4 kinds of nitrogenous bases. That is adenine, A, thymine, T, guanine, C, and cytosine. So, it is the arrangement of these nitrogenous bases that arranges to form genetic code that codes for amino acids. Now, how these bases gets arranged to form a codon that codes for a genetic code? If we suppose that these 4 nitrogenous bases occurs in single manner, that is forming singlet codon, that is A, T, G and C. So on this basis only 4 codons would form, but the coding of 20 amino acids should occur. On this basis 4 codons are very less. So the second probability that was taken was of doublet codon. That is A, T, G and C on multiplication with the same bases, that is A, T, G, C. So the probable number of codons that becomes arranged by the multiplication of the two would be about 4 into 4, that is 16 codons. Such 16 codons would also be insufficient for coding 20 amino acids. So, later in 1954, a scientist named Kemov suggested that the codon arrangement within the amino acid should be arranged in a triple code manner. That is, should be in the form of triplet codon. That is, the 4 nitrogenous bases multiplied with 4, again with 4, which forms a triplet codon that results into 64 codons. Thus, on this probability, the number of 64 codons that were formed were sufficiently more than enough to code 20 amino acids. And so, this rule of triplet codon was taken into consideration to understand the genetic code and its coding for amino acids. This figure depicts the triplet codon of m-RNA for amino acid that is represented in a tabular form. As it can be seen from the figure, here the 4 nitrogenous bases are arranged in 3 manners. which on multiplication with each other forms 64 codons, that is each represented for a single amino acid. There are a main properties of genetic code. So let us understand few of them. That is, these properties forms the base of any genetic code for the synthesis of amino acid, that is protein. First, genetic code should be of universal type of code. That is, the arrangement of 3 codons should be specific for any type of amino acid. That is, this universal type of code determines the same amino acid in all organisms which includes virus, bacteria, plants, animals, that is all organisms. The second type is, that is the second property is, the genetic code should be specific in nature. That is, any one codon should specify the position of one kind of amino acid only. Third, the synthesis of a polypeptide chain should be initiated by a codon which is defined as initiation codon. Further, as said earlier, that the genetic code occurs as a triplet codon. Out of which 61 codons codes for amino acid and the rest 3 do not code for any acid. Thus, out of 64, only 61 codons are identified, that is, identifies a typical amino acid, while the rest 3 do not code for any amino acid. Thus, a single amino acid may be specified by many codons, as we have already seen in the table. Such codons are defined as degenerate codons. Out of these all codons, the codon AUG, that is the triplet one, possess dual function. That is, it is considered as the initiation codon for the synthesis of any amino acid, thereby protein, and it codes for amino acid methionine. Thus, it is considered as the initiating amino acid. Further, there are also termination codons, that is UAA, UGA and UAG. Such codons are called termination codons, where the synthesis of protein acid gets stopped. And therefore, they indicate the termination at the synthesis of protein. These codons do not code for any amino acid, unlike initiation codon which codes for methionine Hence, these codons are called nonsense codons. Because they do not code for any amino acid. Then, last but not least, the sequence of genetic code and amino acid within a protein molecule defines or shows linear parallelism. That is, a straight and parallel nature of the genetic code and amino acid within a protein is determined with the help of its sequence. Apart from this, there occurs a point mutation that is associated with the genetic code, which results in many diseases like sickle cell anemia. This is a disease which occurs due to the point mutation that occurs in the genetic code. And so such a mutation can also be called as gene mutation.